Hi everyone, I'm Aldo Pastore, bioinformatics researcher at Fondazione Pisana per la Scienza, a research foundation in Pisa. And the project I'm gonna present you is named Decoding tumor-educated platelets in pancreatic ductal adenocarcinoma, integrating mutational analysis with machine learning for early diagnosis. The context of the project is the PDAC tumor that is a very aggressive disease with a 5 years overall survival of just 13%. This lethality is due to the fact that the only effective treatment is the resection, but only 20% of the patients are diagnosed in advance when the disease is resectable. To diagnose in advance the disease, we need biomarkers, but unfortunately the only available at the moment is the CA19 that has several issues, like the fact that this protein is not expressed in the very early stages of the disease. So in our project we searched for a new source of biomarkers, and we selected a very promising one, the tumor-educated platelets, that are basically platelets that have been modified in terms of phenotype and RNA content by the cancer cells. We exploited a large dataset involving more than 400 samples, including both PDAC and healthy donors, but also non-malignant patients, including pretumoral lesions such as IPMN. Our datasets involved the RNA of the platelets, and on this RNA we called the variants with a pipeline tuned for the variant calling for RNA-seq. We extracted the mutation in all the groups, so PDAC, non-malignant and healthy, and we found mutation in all of them, but of course the highest rate of mutations were found in PDAC. Subsequently, we searched for genes that resulted significantly more mutated against the healthy donors in the same time in PDAC and in non-malignant samples by means of shared differentially mutated genes between PDAC and non-malignant. And we found a set of more than 140 genes. On this set, on the mutation of these genes, we applied a clustering algorithm, and we found two main clusters of mutated genes that then we proceeded to characterize. The genes, after characterization with protein and RNA-seq external database and with pathway analysis, resulted linked to pancreatic cancer and cancer in general, the first, and the second instead to the activation of the platelet, that resulted an important hint respect to the phenomenon that we were investigating, so the tumor education of the platelets. The next step was to select a set of potential biomarkers from these two main clusters of shared mutated genes. And we selected these genes as the genes that showed the highest rate of mutation, so more than the median, but also that show a very interesting feature, so the sharing of the mutational sites, at least one mutational site, between the PDAC and non-malignant populations, as the SLT1 gene that is showed in the slide. Finally, because of the fact that platelets have an efficient splicing machinery, we thought that more information could have been found in the isoforms. So, exploiting the potential biomarkers that we identified with mutational analysis, we extracted the isoform from them. And with the isoforms, we built a machine learning model using the XGBoost classifier. The model basically discriminated between the PDAC and pretumoral population against the healthy donors in a early diagnosis fashion. The performance of our isoform-based predictive model are shown in the slides, and specifically resulted in very good accuracy both when considering the non-malignant against LT donors and the PDAC against LT donors conditions, and of course in the full PDAC plus NM against LT donors. So to sum up, we basically analyzed the RNA-seq of the platelets to find novel source of biomarkers for PDAC early detection, and we found two main clusters of mutated genes linked to cancer and to the activation and education of the platelets. Then from these two clusters we extracted a set of potential biomarkers with the interesting feature of sharing a mutational sites between the PDAC and non-malignant population. And from them we extracted the isoform and we built a predictive model capable to discriminate LT donors against the disease, pancreatic disease condition in a early diagnosis fashion. And finally and importantly, we are now running the in vitro validation culturing platelet and PDAC cancer cell in order to examine in deep the uptake of tumoral molecules from platelets. Thank you.